Good morning, everyone. Welcome to today's webinar hosted by ACD. As we are waiting for people to join in, I would like to give everyone just a few more minutes to ensure that you are logged in and have checked your audio. Webinar will begin shortly. Once you have joined the webinar, please make sure the audio is connected by choosing the right option. If you decide to call in, the call-in numbers are listed on the right side of the slide. Thank you. Thank you for your patience, everyone. Before we begin, I would like to let everyone know that the audience will be automatically muted for the duration of the webinar. If you have a question, please use the Q&A function to the right of the WebEx window. Please address your question to all panelists, and my colleagues will respond to your queries in real time. In the event we are unable to get to your question, we will follow up with you offline after the webinar. This webinar is being recorded and will be on our website soon. Here is a quick overview of the topics I will be covering today. I will begin with an overview of ACD's in situ hybridization technology, followed by the general workflow and the sample preparation guidelines. Next, I will discuss BaseScope assay workflow, problem points, troubleshooting tips, and also the various application areas of this technology. Towards the end, we will go over a few frequently asked questions. So let's start today's discussion by understanding the RNAscope technology. The RNAscope assay is an in situ hybridization technique that utilizes a unique double Z target probe design followed by a signal amplification system which allows you to visualize a single mRNA transcript as a punctate dot through either chromogenic or fluorescent detection. This highly specialized system utilizes simultaneous signal amplification and background suppression methodology. RNAscope is compatible with a wide range of sample types, such as SSPE, fixed frozen, as well as fresh frozen. RNAscope can be used to detect multiple mRNA targets simultaneously in a single tissue section. Here is a brief overview of the RNAscope workflow. RNAscope assay is a slide-based assay and is intended for use on sample types similar to that used with IHC or IF. The assay starts with permeabilizing the sample with reagents provided in the pretreatment kit. The pretreatment allows unmasking of the target mRNAs, thus allowing the target probe to penetrate into the cell and hybridize the target RNA sequence. The target probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. A series of amplification cascade can then amplify the signal, and mRNA molecules can then be visualized under the microscope as punctate dots. The signal can also be quantified semi-quantitatively or quantitatively using image analysis software. The primary feature of our RNAscope technology is the probe design. Represented here are the oligonucleotide target-specific probes referred to as double Zs. They have two unique regions connected. This allows, to buy a linker. Additionally, each one of these oligonucleotide sequences have been designed using an informatics algorithm which selects sequences that specifically bind to the target sequence of interest. This design confirms that the oligonucleotide sequences do not cross-hybridize with any other sequences. The sequence at the bottom of the Z is complementary to the target transcript. In order for the amplification to occur, two Zs must hybridize to the target sequence right next to each other. Once this happens, it creates a 50-base target-specific binding site on the bottom of the ZZ pair. The top of the ZZ is the base for the amplification structure. When two Zs hybridize, it creates a binding site upon which a pre-amplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of 1,000 bases or more consists of about 20 ZZ pairs pooled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind the target RNA to generate enough signal for molecule detection. Here is the schematic showing the hybridization and the subsequent amplification of the RNAscope assay. The double ZZ probe will bind to the target mRNA transcript. Once the double Z probe binds, it serves as a foundation and binding site for the preamplifier molecule. Once the preamplifier is bound, it serves as a binding site for the amplifier molecules, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each ZZ binding site. Labeled probes can contain a chromogenic enzyme, such as HRP, that generates a visible signal after chromogenic reaction, such as with DAB, detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under a fluorescent microscope. This signal amplification strategy provides for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule. If a single Z binds to the target mRNA transcript, the preamplifier cannot bind as it needs both ZZs to provide a binding site. This helps to ensure the specificity of the probes and helps reduce potential background staining. In order to detect multiple mRNAs simultaneously, multiple amplification trees must be built. That being said, we must ensure specificity. The specificity is ensured by adding a unique tail sequence to the probes. This means in the double Z probe design, we have a tail sequence that is indicative of a channel designator. For example, a C1 probe that has a specific C1 tail sequence that will only allow for binding of the subsequent C1 and preamplifier, amplifier, and labeled probe molecules. Similarly, a C2 probe will have a separate and unique tail sequence. This slide summarizes all the assays that are currently available from ACD. We have a chromogenic singleplex, brown or red assay, and a duplex assay on both manual and automated platform. The multiplex fluorescent assay allows detection of up to 3 targets for V1 assay, or 4 targets for V2 assay, or with the high-plex kit, up to 8 or 12 targets simultaneously in a single tissue section. The major difference between the V1 and V2 fluorescent assay is that The V2 assay uses opal dyes from Akoya Biosciences for signal visualization and provides a high signal-to-noise ratio, whereas in the V1 assay as well as the high-plex assay, the fluorophores are conjugated and are included in the assay kit. If your target of interest is shorter than 300 base pairs, it is below the RNAscope detection limit, and in such situations, BaseScope would be the right choice for you. Currently, BaseScope Red assay is available on both manual and automation platforms. If you are interested in the automation platform, please contact your local account manager. We can have our local field application specialists available for training and troubleshooting on the automation platform. Let's now come to the core of today's webinar topic, the BaseScope assay. BaseScope is the next generation in situ hybridization. It is based on the RNAscope technology, but there are several major differences. First, a standard RNAscope probe contains 20 ZZ pairs, which targets a 1 kb region of the target sequence, whereas BaseScope can detect a signal with as low as 1 ZZ pair probe. Both RNAscope and BaseScope are capable of single molecule detection, meaning that a single dot represents one single RNA molecule or target. RNAscope is a robust assay to detect gene expression of most RNA targets, whereas BaseScope is specifically designed for the detection of splice variants, short target sequences, and point mutations, which I will describe in further detail later during this presentation. Further, RNAscope is able to detect up to 3, 4, or even 12 targets simultaneously, whereas currently BaseScope can detect 1 or 2 targets. Detection with RNAscope can be chromogenic or fluorescent, whereas BaseScope is available as a chromogenic single-plex assay with fast red detection and as a chromogenic duplex assay with fast red and green chromogen detection. RNAscope can be performed manually or by automation, whereas currently only BaseScope single-plex is available on automation platform. Please note that BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents, and vice versa. RNAscope probes too cannot be combined with BaseScope reagents. The assay workflow for BaseScope is very similar to RNAscope. Based on our experience, BaseScope assays may require a stronger pretreatment due to the additional amplification step required. As mentioned earlier, do not mix the RNAscope and BaseScope reagents as they are not compatible with each other. Here is the schematic depicting the chemistry of BaseScope single-plex assay. The amplification tree that is built for the C1 probe is based on alkaline phosphatase chemistry and visualized using the fast red chromogen. Please note that fast red chromogen naturally fluoresces at 570 nanometer wavelength, and therefore the red signals may be visualized under a fluorescent microscope as well. This is an overview of the BaseScope single-plex red chromogenic assay workflow using FFPE samples. As in RNAscope, BaseScope assay also has 4 major steps: pretreatment, hybridization, amplification, and finally, staining and detection. The assay usually takes about 11 hours and can be split between 2 days. Please refer to Part 1 user manual for sample preparation and pretreatment recommendations for your specific sample type. Pretreatment step is key in getting successful staining, so please make sure to follow the protocol exactly as described. Now here's the schematic depicting the chemistries of the duplex assay. Here you can see that there are 2 independent trees that are built for each channel. For the C2 tree, it is the ALP, and for the C1 tree, it is the green substrate using the HRP enzyme. Please note that C2 probes are always provided in a 50x concentration and are required to be diluted prior to the assay. This is an overview of the duplex chromogenic assay workflow using FFPE sample. The broader steps remain the same and include pretreatment, hybridization, amplification, and staining and detection. Here, there are a few additional amplification steps as you now have to detect both the ALP-based fast red color as well as the HRP-based green chromogen. Let's now take a look at a few things that you would be needing to run a BaseScope assay. This is the standard RNAscope kit configuration, which includes the necessary pretreatment reagents, the detection kit of choice, and wash buffer. The probes are not shown here. Shown here are some recommended accessories required for running the manual RNAscope and BaseScope assays. At the top left are the two options for washing and handling slides. You may use either the TissueTech wash tray, common in most labs, or you can use our Easy Batch slide processing system, which was designed to streamline the RNAscope and BaseScope assays and save handling time when working with a large number of samples. The high-busy hybridization system to the right is designed to maintain the required temperature and humidity to ensure optimum and consistent RNAscope-BaseScope staining. In the bottom row are two methods for carrying out the heat-mediated targeted retrieval step required for some sample types, such as FFPE and fixed frozen. Our website has training videos on the different steps of the manual assay, which is a great visual guide to assist with running the RNAscope and BaseScope manual assays. On this checklist are some of the key components that can help ensure your success with the RNAscope assay. For example, using SuperFrost plus slides only, using high-busy hybridization system, etc. We also have some recommended general guidelines, such as once you begin the assay, do not let the slides dry out, warm probes and wash buffer at 40 degrees centigrade in case you see precipitation, etc. Some general tips to ensure success with RNAscope assay are, first, both RNAscope and BaseScope assays, actually. First, follow the protocols. We have various protocols with necessary details to help address the best way to run the assay. Next, sample pretreatment is very important to ensure that you get the optimum assay signal. The pretreatment conditions will differ from sample to sample type and may require some optimization. Using controls is highly recommended, especially when you are troubleshooting your assay. And lastly, it is critical to make sure that you have all the required components before you begin the assay. Here is an example of the recommended sample separation. With FFPE samples, we recommend fixing with 10% NDF for 16 to 32 hours at RT. The recommended sample thickness is 4 to 6 microns, and we also recommend that the tissue is mounted onto SuperFrost plus slides. In order to preserve RNA quality, it is recommended to store sections with desiccants at room temperature for up to 3 months. For long-term storage, it is best to store at 4 degrees with desiccant. For fresh frozen samples, please review the user manual for detailed recommendations. A few highlights are presented here. We recommend storing frozen tissue blocks or sections in airtight containers at -80 degrees centigrade for up to 3 months. The recommended sample thickness is 10 to 20 microns, again mounted on SuperFrost plus slides. BaseScope is a highly specific and sensitive in situ nucleic acid hybridization assay and requires that the protocol be followed closely. To ensure first time and also continued success in the specific detection of your intended target, we strongly encourage all users to perform a technical quality control to ensure that the assay is being run appropriately. This technical control is easily done using a cell pellet control sample tested on 2 slides with a medium copy housekeeping gene PPIB positive control probe and a non-specific bacterial negative control probe. With proper execution of the assay, results will show strong positive control probe staining and clean negative control probe staining. it is recommended that you always run a pilot assay with the control cell pellet slide to verify that the assay technique is correct. Once you confirm that the assay is working as expected using the control cell pellet slide, you can next use the same negative control DAPB control probe and positive PPRB control probe on your experimental tissue to verify the sample RNA quality and optimize the pretreatment conditions. Similarly, for BaseScope duplex assay, you will run the QC with positive control probes for two housekeeping genes, low expressing POLR2A and moderately expressing PPIB. If your sample passes QC, you can then move on to running your target probe. Otherwise, it is recommended to verify your technique, check RNA quality, or perform further assay optimizations. Coming to BaseScope data analysis, BaseScope signal appears as numerous punctate dots, either red for the BaseScope red chromogenic assay or green and red for the duplex assay. These dots are of various sizes and intensities, and sometimes a few clusters may also be seen. Typically, it is the number of dots and clusters that are important rather than the intensity of the dot. Each dot represents one mRNA molecule. When multiple mRNA molecules are present, the signal appears as a cluster. The BaseScope staining can be quantified by manually counting the number of dots per cell. This is also known as the semi-quantitative scoring method. With this method, a score of 0 to 4 is assigned based on the average number of dots per cell. We have a guideline document on our website that you can download that describes the different methodologies to quantify your results. If you have further questions, please feel free to contact technical support for recommendations. Let's now review some troubleshooting examples and results after performing optimization on these samples. When running the RNAscope assays, there are a few factors that could affect the outcome of your results. For example, fixation conditions are not optimal, or hybridization conditions are not optimal. These factors and issues can be addressed by the following solutions, such as the one listed here. Fix the samples as recommended. Use the HiBZ hybridization system for optimal hybridization conditions. One of the common issues that we see is related to tissue adherence to the slice. To troubleshoot sample detachment, we recommend SuperFrost plus slice to ensure optimum adherence. If you are observing tissue detachment, the likely causes are listed here. We have found that including an additional baking step can mitigate this issue. I will now review in detail the additional baking steps incorporated for FFPE and also fixed and fresh frozen samples. These apply to all assay types. For an FFPE sample, if you're observing detachment after hydrogen peroxide or target retrieval, then add a baking step before the hydrogen peroxide step. Or if you're observing tissue coming off from the slice after protease treatment, add an additional baking step before the protease step. Both the baking steps are for 30 minutes at 60 degrees centigrade in a circulating dry oven. For sample detachment issues with fixed frozen samples, we recommend drying the slice at 20 degrees centigrade for 2 hours after cryo-sectioning and adding a post-fixation step before the hydrogen peroxide step for 15 minutes to 1 hour at 4 degrees centigrade. We have found that majority of the sample detachment happens after the target retrieval step, and so we recommend adding an additional baking step before the protease. After trying the steps 1 to 3, if you continue to observe tissue detachment, then we recommend that you omit the target retrieval step and extend the protease time to 45 minutes. You can also contact support and we can help you figure out the best troubleshooting path for this issue. For sample detachment issues with fresh frozen tissues, we recommend extending the post-fix duration to 1 hour at 4 degrees centigrade and also include a baking step at 37 degrees centigrade for 30 minutes. Let's now take a look at some of the applications of the BaseScope assay. The BaseScope assay has been used in several application areas. The most popular research areas include oncology and neuroscience. Since point mutations and splice variants have significant pathological implications in cancer, BaseScope is frequently used to visualize these targets in situ. Similarly, we have seen wide application of the BaseScope assay for detecting circular RNAs, gene fusions, and splice variants in the brain. Target-specific applications of BaseScope can be divided into three major areas. Exon junction probe design can be used for the detection of splice variants, isoforms, circular RNA gene fusions, and confirmation of gene knockouts. These probes are all 1 ZZ in length. Short target sequence probe design can be used for sequences between 50 and 300 nucleotides, highly homologous sequences, CDR3 sequences for T-cell receptor clones, precursor microRNA, microexons, small nucleolar RNA, and gene editing. These probes can be 1 to 3 ZZ in length. Lastly, we have a list of in-house validated point mutation probes that can be used to identify specific point mutations in cell and tissue samples. An example of the application of BaseScope for exon junction detection is the variant MET-delta-14. The MET-delta-14 transcript lacks the exon 14 of the MET gene. This transcript is frequently observed in advanced lung cancers. Exon 14 skipping of the MET gene occurs due to an aberrant splicing event. In a normal splicing event, introns from the pre-mRNA get spliced to generate a coding mRNA transcript. In case of the aberrant splicing event, exon 14 gets spliced out along with the introns, leading to a coding mRNA transcript without exon 14. This leads to a functional change in the MET protein, leading to increased cancer cell growth and survival. With BaseScope assay, probes designed to span specific exon junctions can identify the wild type and delta 14 MET transcripts. Probe designed against exon junction 12 to 13 will recognize both the wild type and variant transcripts. Probe designed against exon junction 14-15 will only recognize the wild type transcript, while probe designed against exon junction 13 to 15 will only recognize the mutant transcript. Another study was done on H596 cell lines that has the MET variant, whereas the A549 cells have the wild type MET transcript. The variant-specific probe shows positive signal only in the H596 cells and not in the A549 cells. Similarly, the wild-type specific probe only detects signal in the A549 cells. While the probe designed against the common region of both the wild-type and variant transcript show positive signal in both cell lines, the probe for DAB shows no signal, indicating no background. This demonstrates the specificity of the BaseScope probe in detecting specific splice variants. BaseScope duplex assay has been used to detect CRISPR-mediated nucleotide deletion. CRISPR-Cas9 was used to delete 49 nucleotides from a gene, resulting in a novel junction sequence labeled edited. BaseScope 1ZZ probes were designed to target either the 49 nucleotides in the wild-type sequence or the edited novel junction sequence. Using the BaseScope duplex assay, the wild-type and edited transcripts can be visualized simultaneously. The image on the right shows staining of wild-type biallelic or monoallelic hepatocytes. Another application area is the visualization of point mutation. BaseScope assay can detect transcripts down to a single nucleotide change. For example, KRAS G12C mutation is observed in several malignancies, predominantly in lung tumors and colorectal cancer. It is a driver mutation which promotes tumor growth and results in resistance to standard therapies. Using 1ZZ point mutation probes targeting either the KRAS G12C mutated sequence or KRAS G12 wild-type sequence, it was possible to visualize the positive signal in cells homozygous for the KRAS G12C mutation. BaseScope can also be performed in combination with immunofluorescence. This natural complementarity of these assays allows for the detection of RNA and protein targets of interest on the same sample section. The BaseScope assay is performed first, followed by IHC or IF assay. A nice example of the dual BaseScope IHC assay is the visualization of the DL gap transcript within the dendrites of the neuronal cell. The DL gap circular RNA can be observed in red, while the MAP2 antibody specifically marks the dendrites. The same section was stained with DAPI and imaged using a fluorescence microscope. As mentioned earlier, the fast red from the BaseScope signal fluoresces red, and a fluorescent labeled antibody was used for the detection of MAP2. This provides a nice contrast and more distinct visualization of the DL-gap1 circular RNA within the dendrites. A preliminary feasibility study by Wu et al. Used BaseScope assay to detect EGFR T790M mutation in formalin-fixed paraffin-embedded tissues of patients with lung cancer. This was published in Thoracic Cancer Journal in 2019. EGFR is a prevalent mutation in lung cancer tumors and promotes cell growth and progression. EGFR tyrosine kinase inhibitor treatment is used as targeted therapy for patients with EGFR L858R mutations, while patients with resistant tumors often develop EGFR T790M mutations, which respond to third-generation tyrosine kinase inhibitors. The BaseScope assay was successfully able to detect EGFR T790M and EGFR L858R mutations in patient samples with high sensitivity comparable to PCR along with spatial and histological context. In another publication, Volpe et al. Used BaseScope in situ hybridization assay to discriminate D16HER2 variant expression from the wild-type HER2 isoform in breast, gastric, and colorectal cancers. The HER2 splice variant D16HER2 is characterized by the deletion of exon 16 and is associated with HER2-positive breast cancer aggressiveness. With the exception of qRT-PCR, easily reproducible assays are still lacking to clinically detect and quantify the D16HER2 expression and in the tissue context. BaseScope assay has been used successfully in this scenario. Johnson et al. Used BaseScope IHC to detect a subpopulation of T cells that predominantly contributed to anti-PD-1 immunotherapy-induced encephalitis in a patient. The BaseScope IHC workflow allowed validation and visualization of the clonal T cells identified by PCR sequencing. On the new product side, we now have a new assay platform to detect microRNAs, antisense oligonucleotides, and small interfering RNAs called microRNA Scope that will be in market soon. MicroRNA Scope is a robust assay offering high sensitivity and high specificity with a universal probe design. The custom probe is designed to detect microRNA, antisense oligonucleotides, or small interfering RNA sequences of interest. The microRNA Scope assay can be combined with downstream protein detection and is compatible with FFPE, fresh, fixed frozen, and cultured cell samples. It will initially be available as a single-plex chromogenic fast-read assay, both manual and on Leica automation. Publications are a significant piece of scientific materials that researchers leverage for understanding current trends. ACD has over 2,600 publications as till date. Within the thousands of publications, BaseScope has over 80 publications in just 4 years. A fun fact to know is that an average of 25% of our publications are published in Nature, Cell and Science, the top 3 scientific journals out there. For more information on our product, please visit our website at acdbio.com. If you click on the Support tab, we have a wealth of information including troubleshooting guides, user manuals, tech notes, training videos for the manual assay, as well as previously recorded webinars that cover a wide range of different topics and applications. For those of you who are just getting started with ACD's assays, we offer a new user program where we pair you up with one of our technical support experts to provide guidance throughout your experiment and ensure a successful run. For those of you who would like to send in samples and prefer the assays to be validated by ACD, we offer pharma assay services or academic support services. Our services team ensures all due care is taken with your samples and that the assay development and analysis are conducted with highest efficiency. With that, I would like to thank everyone for attending today's webinar. In just a couple of more minutes, I will post some of the most frequently asked questions. My colleagues are still on the line to help address your questions. So please feel free to continue sending your questions. Again, as a reminder, this webinar is recorded and will be available on the website. In the event you were unable to get a question sent through the Q&A feature, please find the contact information for technical support on this slide, and you can email us your questions.